Hi, BBSOAS community. Current research efforts into BBSOAS and the NR2F1 gene are underway in Germany with Dr. Shah at Heidelberg University, in France with Dr. Studer at Nice University, and Italy with Dr. Silvia De Marchis at the University of Turin, all to better understand the NR2F1 gene and ultimately identify treatment. But here's the missing piece. During our October 2025 strategic research meeting, our scientific advisory board and our research council were absolutely clear. In addition to the ongoing work in Europe, we need a US-based research lab to accelerate discovery and development of targeted treatments for BBSOES. Building on all of our projects to date, we are taking the next critical step and creating a US-based BBSOES research lab. I'm really excited to have Dr. Veeral Shah with us. We need to raise $300,000 over the next 3 years, so $100,000 per year, to establish the first dedicated BBSOAS research laboratory in the United States at Cincinnati Children's Hospital Medical Center, led by Dr. Veeral Shah. Dr. Shah is unique as a pediatric neuro-ophthalmologist. He's the only one in the Midwest. He's been part of BBSOAS since his time at Baylor, and he was involved in our first NR2F1 family conference in 2018 that inspired the creation of the foundation. But what everyone might not know is that he's also got a big background in basic science, which means training and understanding how things work at the most fundamental level, like studying cells, genes, and biological processes in the laboratory before applying that knowledge to treating patients. Dr. Shah, let's tell the community more about you and this exciting project. Can you tell me how you first got involved with researching BBSOES?
Yeah. So I was in at that time I was a pediatric neuro-ophthalmologist. Started out of training. I started at Texas Children's Hospital. I was in Houston, Texas. Great environment. Got to see a lot of the huge. I think the largest medical complex in the world is. They tout that. They do everything big in Texas. But at Texas Children's, I was affiliated with Baylor College of Medicine. I had a basic science lab there. I did research. I was involved in clinical research, and where there's a lot of amazing genetic research, and this is where the NR2F1 gene was first identified. Whether multiple members, but one of the founding members for the BBSOAS was Christian Schaff, and he and organized the group and the patients that he was seeing to her patient conference. And that happened, and I believe in 2018, when we had that conference. And so that was just a wonderful experience that brought these families from around the world here to Houston, Texas, for the first time to really understand and learn. And he had conferences, and we actually had an opportunity to do clinical evaluations and get everyone sort of up to par about thinking about this gene and really learning. We were more so learning from each other, learning what other families were learning from other families about their children and their child with NR2F1 and BBSOAS. And so that was a wonderful opportunity for myself and my colleague at that time, Jane Edmond. We were two pediatric neuro-ophthalmologists. And there are not many of us. There's like maybe like 20 or 25 of us in the nation. So to have two in Texas, together, we had the opportunity to, see the families and actually see the children and do a really high evaluation and neuro-ophthalmology evaluation, and it revealed a lot to us about vision development and NR2F1 and BBSOAS.
Mhm. So, how does your background in basic science inform the way that you approach clinical care with your patients?
Yeah, so as a pediatric neuro-ophthalmologist, so clinically, let me talk about it clinically, and then I'll talk about the basic science. So clinically, what I do is where ophthalmologists feel uncomfortable about what's going on behind the eyes, and neurologists feel uncomfortable about what's going in the eyes. A pediatric neuro-ophthalmologist is that connection between the two. So really, with the central nervous system and the eye and how that works, and much of our brain, much of the human brain is devoted to vision. And so I found it early in my career that I enjoyed understanding, seeing the challenges that we see both in pathology and misunderstandings of what's going on with our diseases involved in the brain. And so early on, I started doing, I got involved in training with research. So I'm an MD, PhD. I did, 2 years of medical school. I did an entire PhD. I looked at animal models. So I looked at both chick development, but also mouse development, and looking at particular genes and how they work and understanding their function in the animal models. Basic science is a wonderful way to understand how a gene or how a protein affects development and what are the outcomes, and also what things we can do to answer when those proteins are not being expressed or the genes are not being expressed, and really think about solutions into fixing errors in development. And the animal models allow us to really answer some of those questions and understand those relationships. So, what I love about what I do, and I really enjoy, and I have a lot of passion for pediatric neuro-ophthalmology, is that some of the clinical questions that I see and the complicated stuff that I see in my clinical lab, it drives me to go back to answer some of these questions by developing animal models for these issues, and then understanding that relationship at the level of the animal models, and then translating back those relationships back to understanding what potential interventions or human therapies we could design. And I think that's where I get a lot of passion and a lot of drive for being involved in both, just as a clinician, clinical research, but also the basic science, research of it. So my background, I've done a PhD both in animal work. I've done a postdoc in animal research, and I continue to do it at many levels in different neuro-ophthalmic issues that we see that affect vision outcomes.
And so in clinic, how many BBSOAS patients have you seen to date, and what have you learned from caring for these families?
Yeah, so I've seen, I looked at it the other day because I had to put together my list, and it was we have 44, that I've
Wow.
and that's a number of kids, and more than half of them, more like 60% of them I'm seeing on follow-up, meaning or continuing care and seeing them. The other one third, I think the thought is the follow-up, but the question is distance and time. And we're seeing a number of people from international locations, South America, from India, Europe, and to name a couple that are coming to see us, and so it's always a hard, but we're in constant communication. So if I say, I think that number is closer to 90% if we talk about communication and talking and about what things, how we're helping them still outside that initial evaluation. But the
Yeah.
evaluation that we get is quite comprehensive, and it's a neuro-ophthalmic. It's not a general ophthalmology evaluation. It's and so your first part of that question was how many, but then the second part was what have I learned, and what I've seen is that it is true to its nature. It's a spectrum disorder, and I think it's every child is different, just which is true without talking about NR2F1 or BBSOS. But when you have an issue like NR2F1 or BBSOS, how each child deals with it or collectively is different for them. And so the treatment or or what ways we can intervene or adaptations for that child to improve their day-to-day living and functioning and live at a healthier state of well-being really needs to be catered to that child. And so the evaluations really have to be catered to really pick up those impairments to what we need to focus on. And so what I've seen is I've seen kids with cerebral visual impairment. I've seen kids with behavioral issues or autism. I've seen people with learning disabilities, and I've seen that where it's some combination of those three, or a lack of combination of those three. So some, a derivative of those things. So I think it's really important to not do one size, fits all
Mhm.
therapy or interventions. It's really about understanding what you're seeing and what things can optimize, that child. And we've done things where we've done surgery on certain kids, which have helped their nystagmus, their head position, and you've seen their development take off. within the group. You've seen people where we've done significant understanding from the parents of what's going on. We've had interventions from CVI interventions where they've seen a progression in their vision development where there wasn't one before. So I think it's been great so far. And I think what I've learned more than anything is that no child is the same. Every child is unique and different, and we approach that evaluation as such.
Mhm. So when we think about sponsoring a postdoc researcher and creating the clinic for you at Cincinnati Children's Hospital, what makes Cincinnati Children's Hospital uniquely positioned to advance research in BBSOS? And can you share
Yeah.
what specialities and resources you have to access that will make a difference for our patients?
Yeah, of course. Yeah. Um, um, so Cincinnati Children's Hospital, I left Texas Children's Hospital to come to Cincinnati Children's Hospital, and what I've been amazed, first off, all is the infrastructure for research. I mean, so Cincinnati Children's Hospital, when I came to Cincinnati, was number three, and since then has been the last several years has been ranked the number one children's hospital in America. So the facilities are amazing. Cincinnati itself is a smaller city compared to some of the other cities, but where it has benefited from that is that it has a huge encaption area and it really covers the Midwest. So the patients any given day are from many different states that we see in its location where Cincinnati is. But what it has done is invested its money really well in establishing an amazing research center. I mean, I don't I've trained at other top children's hospitals, training both at residency and at fellowship level. I've visited them, but the research opportunities are amazing here. So I think where it's elevated is that for animal work, let's say they have animal facilities, multiple animal facilities. They have core facilities which allow you to process tissue quicker, which are all up to date, which are state-of-the-art technology for genetic assessment, protein assessment, really about biostatistical, and which is very much needed for genomic analysis. And then tissue, as I mentioned, tissue processing, but also like animal care, right? And animal behavioral work, which is so key to what we need with the animal models that we have with BBSOAS and NR2F1. I'm sorry, I know it's a long-winded question, but it's the number one children's hospital. And more than anything, some of the facilities from a clinical standpoint, um, um, you won't find children's hospital which are insular to the children's hospital. So the CVI team that we have here is probably one of the longest standing CVI teams in the nation. I think it's one of the best teams in the nation. It's insular to the hospital, so all the care is not fractured like it is in other places where you have the diagnosis, go get, PT here, get OT here, get this service here, and they're not communicating. So what gives it best for the child? Here we're all together, the PT, OT, we're all within the same center, the Perelman Center. We're all communicating and talking. We're getting the best outcome for that child, and we're all trying to figure out what we can treat that child holistically. So I think that being a long-standing 10-year, institution with infrastructure is amazing. And I think that's what makes Cincinnati Children's unique in terms for addressing vision-related issues, especially CVI, even some of the behavioral and development issues as well.
Mhm. So if we're thinking about the postdoctoral researcher, and we're obviously asking our community support to make this possible, tell us more about what the postdoc researcher might actually be doing on a day-to-day basis under your supervision, and what aspects of BBSOS and NR2F1 would they be focusing on?
Yeah. So I think majority of that time would be involved in basic science, research. I think it would be a combination of basic science and clinical research. And I would almost be, I don't want to put percentages on it, but I would like 80% of the time in basic science research because it's going to require that. But the 20% of that time, which is just perfect, will be involved in the clinical aspect of it. And so what we have mentioned previously that I've seen 40 patients of BBSOAS, but I look at my schedule every month. They're either their follow-ups or their new patients coming in almost every month. And so with that, I think one of the goals for the NR2 Foundation is really to consolidate and really understand a registry that would be helpful for all, and for other clinicians and other questions and other things that we come together as a group to say, hey, that's a good question. Maybe we can answer that through our registry. And so I think that post doc would be helpful for that clinical aspect of that research and really the data collection and translating that to a matrix-based system, which I know that's what the NR2 1 Foundation is connected with. Now, the 80%, let me talk about the 80% of it because I'm most excited about that. That's the basic science research. So we have at the moment, I believe 4 models of animal models. I previously, I think 3 or 4 months back, I was at 3 models for NR2F1 animal models, which are amazing because 3 of those models mimic true human,
Mhm.
mutations that we see. And so how that mouse manifests, we've already defined, to some level, how that mouse has manifested similar parallels at what we see in humans. And so they're an excellent model. So not only are they, the mutation is, similar, but what we're seeing as a phenotype is very similar. And so that's a great model system for us to really think about what things we can intervene and interventions. And so one of the thoughts we have, again, NR2F1 is a receptor. We don't know what binds to it, but we have several candidate molecules we know that can bind to it. And going back to BBSOAS, you have two genes, one that works, one that doesn't work. And so the thought has always been if this one doesn't work and this is causing the sort of uh, the spectrum of effects that we see in BBSOAS, if we're able to increase the expression of the good allele, could we mask that or minimize the effect of the pathogenic allele? And that's always been a question. And so, could it be as easy as just having more of an agonist bind to it and increase the activity of the good allele to wash out? And so that's one of our thoughts for intervention. So having these animal models and working through an agonist that could be a potential treatment for our children is a primary goal, not only for the lab, but for the foundation, so.
Yeah. And so for families who aren't so familiar with how academic research works,
Yeah.
can you explain why having a dedicated postdoc for BBSOS will accelerate the pace of discovery compared to research done that's without that focused attention?
Yeah, I think, well, a postdoc day to day, going back to your last question, was day to day, they're going to be involved in understanding what the goals are of the main project is. It's really understanding the the aspects of the model. So even though we have these 3 models, some of the work that's being done right now is understanding what is totally going on with these models, right? And one of the, for example, one of the issues is seizure control and seizures, and where's the nidus for seizure controls? And in these model systems, we haven't assessed these mouse for are they actively having seizures, and if there's a potential treatment to address the seizure aspect of that, in a subset of our kids that we see. So I think, what we're going to do day to day is really learn from the animal models and see how that corresponds to some of the global phenotypes, not only the visual phenotypes, but just the global phenotypes. And the postdoc, him or her, I think they're looking at this with an open onset of looking at systemically what things are going to be better, especially for BBSOAS. It's not going to be just focused on vision, where just me and alone would be focused more on the vision. We're going to totally focus on the vision, but I think it's going to be really holistic, and I think that's going to be really great. And then we're going to not only we have all these amazing core facilities to really assess core behavior, understanding what works and what doesn't work, and then understanding these candidate molecules if they're going to work. And then what we're establishing here is that if we have a pathway that things are looking very successful, let's say a successful intervention or experiments that are looking positive for a candidate molecule, we're having a natural registry, a natural course history registry from the clinical patients we're seeing. I don't think that's happening anywhere else in the world. And as we collect and see more children, we're going to get a 4 or 5 year natural course history that will allow us from a genetic as a drug trial
Mhm.
potential to establish a drug trial to treat children and to see if there's a therapeutic effect, that we can, and having that basic science work to establish you need to have that to get to a point of a drug trial. They don't do drug trials for hope. They do drug trials because they have good science behind it that suggests that hope is really great. Like it's not just the minimal of hope. It's like it's
Yeah.
it's there for us to really test it.
So having this position will really help us as a community to potentially get closer to treatment.
Yeah, yeah. I mean, I
That's
don't know of a complete human treatment for this yet, and I think what the fascinating is why I talked about just agonist and one aspect of genetics. I think the having a postdoc, him or her here, is looking at multiple interventions, not just one. I
yeah.
mean, there's the thought of editing, gene editing. There's different discussions out there about how to approach
Mhm.
gene therapy, and I think a postdoc, him or her focus would be looking at all those avenues, not just
Yeah.
one. Yeah.
Yeah. So until now, our research for BBSOAS and NR2F1 gene has been centered mostly in Europe.
Mhm.
What difference does it make to having a dedicated research hub here in the United States that can partner with our colleagues in Europe as well?
Yeah, I think for one, it's a necessary must. I think we have a large volume of people here in America that are who have this diagnosis, right? And I think it's really important to have it established here. I think two, I you know I Europe is great. America has always had a lot of money invested in research, and I think it has the ability to really drive, really make huge impacts in terms of opportunity, in terms of uh, technology, but also incorporating and funding those endeavors to get to where they need to be. And I think having it established here uh, does many things. One, it allows us to work collaboratively, right, with both groups. It also gives us an opportunity to um, cooperate and among a group and then and consolidate and then work towards solutions together, and I think that's the key aspect of things, and I think that ability to have it at one particular place and have that guidance and have also the ability to do that is unique to America, I
Mhm.
think in many ways.
And at speed as well with our animal models, I'm assuming that we're able to, we don't have as many restrictions in the US as we do in Europe when it comes to animal model testing. So will that
That's
mean
right.
that we'll move quicker?
Yeah, that's right. Yep.
Okay. And so how will you plan to collaborate with our European research team as well to build on the existing knowledge while we advance in hopefully new directions as well?
Yeah, I think they're a partner in this. I don't actually see, in this entirely. So I think it's communication, collaboration, constant monthly meetings, and communicating our findings, understanding their findings, thinking about the next steps. That we're already doing that to some degree. Even from our most recent meeting, I've already had 4 or 5 meetings with our French group and as well as with correspondence with German with Christian and Michelle. We've already emailed and scheduled things. So I think it's this idea that has really been I think one of the profound things that the NR2F1 Foundation has done is really put us together.
Mhm.
And I think we want that. And I think having contributions from everybody and discussing it and collaboration, I think the sky's the limit, I think, and that's where it begins, so I think you've, as a foundation, have really laid great seeds for this, plant to grow and we want to get it to fruition and blossom.
So 5 years down the line, what do you think could look different for families affected by BBSOS?
Yeah, I think 5 years down the line, I think we will get to this diagnosis faster. I think we will have a better understanding of what interventions would be, getting to the diagnosis, understanding that spectrum effect that we've talked about, like where do they fall on classifying these patients, and then addressing what interventions and adaptations they may need at that time. But also getting to the diagnosis is half the battle because really understanding where do they fall in that type of diagnosis. Right now, we're just making a diagnosis of NR2F1, but if it's we're understanding really well what are some of the subtypes within that, and really getting them into the right, treatment and management. And then I think what I see is that kids will be living and being at a better state of being, and in a healthier way of lifestyle for not only for the patient and for the child, but for the family. So.
Mhm. Well, thank you so much. We have got a lot of work to do to bring this to life for you, and we're really excited to come back and speak to you once we have fundraised enough and you've hired the new person, which we anticipate would take about 3 to 4 months. We'll be We'd be really excited to come back and talk to our new postdoc researcher and to hear about what's sort of first on their to-do list to get started. So thank you so much, Dr. Shah, for your continuing commitment to the foundation and really to our children with BBSOAS. And let's see what we can make happen.
Yeah, no, thank you, Jennifer. Thank you to the foundation, and I'm really grateful. I think it's going to be an amazing for the group and for the next steps we're moving forward.
And so for anyone watching who wants to help accelerate research, what would your suggestion be?
Yeah, I would say get involved, whether that's support by showing at the groups or showing at the meetings. It doesn't need to always be financial support, but financial support will definitely help, move things and help us with our goals. That you have a great board, you have an active board. They are very easy to reach and approachable. So when you start to communicate, with them, they're very responsive, and I think, reach out to them, and I think they will guide you to what, supports that you can get involved with. But I think involvement is the main thing, and you can involve in many ways, whether it's just your voice, whether just asking questions, whether that is contributions, voluntary contributions, getting involved in the meetings, but also other ways is being involved in this research is another way you can be in, and financial help would always help too. So.
Mhm. So in practical terms, we families can get involved by taking part in our patient registry, um, uploading their genetic report. So we could sort of look more specifically at the types of mutations. That's something that we've been sort of came out again from our research meeting, patient registry and genetic report. But obviously for this campaign, to bring this postdoc to life, to make all this happen, we need financial support. We can't avoid it. And so whether it's donating yourself or really utilizing your community, so often with living with such a rare disease, other people want to get involved and support. You'll find your neighbors, families, friends, colleagues will say, how can we help? Well, this is a really tangible piece. So you don't have to donate yourself, but as a community, if you can use your network and help fundraise, then that would be fantastic. And obviously from the foundation, we always have volunteer opportunities as well. But getting behind however you can, whether that's your research with patient registry, donating or fundraising would be really helpful.